Hello, everybody. In today's lecture, we're going to discuss the um, physical controls of microbial growth. So let's get right into it. So by the end of this module, you should be able to define the terms optimum, minimum, and maximum as they apply to temperature and pH conditions. You should also be able to describe the temperatures where psychrophiles, psychotrophs, mesophiles, thermophiles, and extreme thermophiles would thrive, and state the grouping for most pathogens. You should also be able to define acidophiles, alkophiles, and neutrophiles, and describe ways that microbes survive in pH extremes. And finally, we should be able to define the term halophile and state how these microbes combat osmotic stress. So let's start out just thinking about how microbes adapt to their various growth conditions. So microbes can live in any environment on the planet, more or less, from our bodies to the soil, uh, into these very extreme environments like the Atacama Desert, where we have some of the driest conditions on the planet, which can increase osmotic stress, or in these hydrothermal vents that are deep in the ocean, which can have extreme temperatures and extreme pressures. They can live in the Great Salt Lake and the Dead Sea, which have uh, different levels of salinity. And then in another lecture, we'll talk about how they live in different oxygen environments as well. So they're just able to adapt to multiple different types of environment. And so we have to think about how they do that. So when we start out, uh, we want to think about temperature effects. Uh, they can affect enzyme activity. So at low temperature, we actually have lower enzyme activity. So this right here is a graph showing the kinetics of enzymes for a specific organism. And we see at the low end, we have low activity. And then at the high end, we also have low activity, and it can come down to 0. And then, of course, there's an optimum level. So this is like the optimal temperature. Now, the reason this exists this way is that enzymes can have, they can actually denature at higher levels. So when we get hot, enzymes denature, they lose activity. But in this, Goldilocks zone, we actually have the highest level of enzyme activity. The enzyme is going to be optimized for that temperature. And this is where you may also see an increase in microbial growth. Now, additionally, we can look at this graph on how temperature affects membrane fluidity. And we see down here at the low end of the temperature, we're going to have a more solid-like membrane structure. This decreases fluidity. And then at the top end, we're going to have a more fluid-like structure to the membrane, and this is going to increase fluidity. Down here on the low end, this decreases permeability in addition to fluidity. Here I accidentally wrote fluidity twice, but the second one's supposed to be permeability. And then up top, as the membrane becomes more fluid, the plasma membrane can eventually disperse because they can't counteract, and then your cell dies. And so again, we have this optimum temperature range where the membrane is at optimum fluidity. So it's somewhere in this range here where the membrane is going to be in its best shape. And so when you think about this, we get it sort of makes sense why you have these 3 different types of temperatures. We have a minimum growth temperature, a maximum growth temperature, and an optimum growth temperature. So they are pretty much what they sound like. So the minimum growth temperature is the minimum temperature at which the organism can grow. And they may be growing very slowly, but they are growing. That means that they are duplicating. As we learned in a previous lecture, we measure microbial growth as cell division, more or less. And then on the maximum, this is the maximum temperature at which they can grow. Now, they may be able to survive below the minimum temperature or survive above the maximum temperature. But when we're thinking about this temperature growth curve, we're thinking about the minimum growth temperature and the maximum growth temperature. And then finally, somewhere in the middle, we have that optimum temperature. This is where they're going to grow the fastest and most efficiently. And so we use temperature and these growth curves as a way to actually classify different microorganisms. And here are two different ways of kind of looking at that. On the low end, we have our psychrophiles. These organisms love cold environments. We have our psychotrophs, which can survive in those cold environments, but their optimum is a little bit higher. We have our mesophiles, which is where most of the organisms that live in the human body are going to be. Thermophiles, which are going to be pretty hot, and then hyperthermophiles, which can be real hot. Excuse me. All right. So your psychrophiles are going to thrive in these cold environments near or to below freezing. Here we have an image of snow algae. My pointer actually looks like another snow algae. But these snow algae are going to actually grow, divide, and metabolize at or below zero in many cases. Their maximum their optimum growth temperature might be above freezing, but they're still having enzymatic activity at like -4, -5 degrees Celsius. Now, your psychotrophs can still grow at these low temperatures, but they don't really like thrive in the low temperatures. More than likely, they're going to have a slightly higher optimum temperature. But these are the sorts of things that you're going to often find in your refrigerator when your food goes bad. And so they're able to grow on, that chicken breast that you left in there for 3 weeks and break down the proteins and the fats. And then when you pop it open, it smells like just open up a garbage can that hasn't been changed for 3 weeks at Taco Bell. So, again, psychrophiles versus psychotrophs, a little bit different. The phials are going to be the ones that really thrive in those low temperatures, and the trophs are the ones that thrive at slightly higher temperatures, but are able to grow down to a pretty low temperature. Then we have our mesophiles. These are usually going to have an optimum temperature in that 30 to 39 degrees Celsius range. So this is the same temperature of our bodies. We're at 37 degrees Celsius. So these organisms are going to include pretty much all the commensal organisms that live in our bodies, as well as all the pathogenic organisms that cause us disease. When we go to our thermophiles, these guys are going to live in these hotter environments. They're going to have optimums well above body temperature, and even their low growth rates are going to likely be above body temperature. So these are highly unlikely to be a human pathogen, although there may be possibilities for that to happen. I'm not aware of any. They can survive pasteurization oftentimes because pasteurization is usually somewhere around 60 or so degrees Celsius. Um, and so these organisms can survive that process. But again, they're usually not of high concern because after we pasteurize, the temperature is going to come down to very low, and that's going to be most likely below their ability to grow. And then if we ingest them, they're not able to grow because we're below their minimum growth temperatures as well. And then we have the hyperthermophiles. These are the bonkers organisms. These things can grow in these extreme environments like this prismatic pond here. And these are oftentimes going to have temperatures above boiling. So, it could be a hydrothermal vent as well. So these organisms are able to grow and divide above boiling point, which is pretty remarkable, in my opinion. But you're definitely not going to find any pathogens here. These things are way hotter temperatures than any living organism is going, outside of bacteria and archaea are going to be able to survive at. Now, one of the things about high temperature environments is that a lot of them end up being like in the deep ocean near these hydrothermal vents. And so organisms that live in these Hot environments oftentimes will also be barophiles. So barophiles are organisms that can withstand high pressure environments of the deep sea. And so what we have over here is Colwellia species in a SEM. And so they're barophilic, which means that they're able to survive at really low temperatures. I think these were actually found in the Marianas Trench. Um, as we go through all this, why does it matter? Well, some of it matters because we're talking about whether or not organisms can cause infection. But other of it is just kind of this interesting result we see where temperature can actually affect phenotype. So if I gave you a image like this, and I gave you, for example, this list of different growth temperature rates, I might, for one, just ask you what kind of organism is this? Is it a psychrophile? Is it a mesophile? So forth and so on. And you could tell that this is a mesophile because of where it grows versus where it doesn't grow. The other cool thing about this is that we see this actual two different phenotypes when it does grow. So at low temperature, these would be like, say, environmental temperatures, we see this kind of dark orange pigmentation. However, when we come closer up to body temperature, we see these more reddish, pink, pale, different colony colors. And so this is the same organism being affected by the temperature that they're being grown at. So when we move on to pH, we also have the same sort of minimum, optimum, maximum relationship. And um, we're going to break these up into just three groups. And it's a little bit easier to remember because acidophiles are going to grow in these acidic environments. Neutrophils are going to be centered around 7, and then alkaphiles are going to be in this like basic environment. And your acidophiles are going to grow in these low pHs that can be at 1 or below, even up to about 5. They're often found in like sulfuric hot springs or volcanic vents. But what's really neat about them is that they usually maintain a neutral cytoplasmic pH. So they're actually able to keep their intracellular pH separate from the extracellular pH. And the way that they do this largely is through the production of proton pumps that can export out all those hydrogen ions that are available in the environment that make it across the plasma membrane. They export them out. This would be a process of active transport, and that's going to help raise the pH. Some organisms can also do things like deaminate amino acids to produce ammonia or produce something like urea. And both ammonia and urea have the ability to neutralize some acids. Although the organisms that do that, I think, are not necessarily acidophilic. They usually do that as a way to deal with with finding themselves in an acidic environment. But those are the different ways that microbes can deal with acids. Now, your neutrophils are going to be right around that central 7, right? That neutral pH. So here we have it listed as around 5 to 8. This is going to be almost like the vast majority of microorganisms, and it's going to be the majority of pathogens. Here we have the pH listed of all these different parts of the body. You see that outside of the stomach, which has a pH somewhere around 1, 1.5 to 3.5, most of the human body maintains a pH quite close to neutral. And so that means that the organisms that live on and in our bodies are also going to maintain a pH quite close to neutral. All right. There are a couple of other exceptions here. I think urine has a slightly lower pH, and the vaginal pH is usually kept slightly low as well. The idea of keeping the vaginal pH low is it helps keep out all those other things that we don't want to cause infection. Then we have our alkalophiles. These are going to grow in these alkaline environments somewhere in the 9 to 11 range. You don't really find many organisms or many environments for that fact that are above 11. Naturally, it's hard to find those environments. While we can find some quite acidic environments, we don't usually find quite as many like pH 14 environments. I'm not sure if those exist. So this might be something like a soda lake, like we see here with all these nice projections out of these like calcium carbonate, most likely projections coming out of the lake. And so these organisms are going to have to deal with an excess pH. And so they're going to need to come close to neutralizing their intracellular pH in the opposite direction. Now, the kind of cool thing about this is that pH is also a major determinant of microbial diversity. So right here, along this y-axis, we actually see microbial diversity. In this case, it's called phylotype richness, but this is just a type of diversity. And along the x-axis, we have our soil pH. And what we see is this kind of unimodal relationship where we get low diversity down near low pHs. Diversity begins to peak around neutral, and as it becomes more basic, it comes back down. And again, notice that in soils, it's kind of hard to find soils that have pHs greater than 9. All right. Then moving on to the last part of this lecture are how do microbes deal with salt? And so most of the bacteria in our bodies are these non-halophilic organisms or non-halophiles. And so basically, they are able to grow in an optimum salt concentration that's pretty low. So here, I think this line is showing you that growth is high, is close to around 0, and like steadily drops. And probably this is maybe like a 1.5% NaCl. Then you have organisms like Staphylococcus aureus that we see with the red line here. These are going to be able to tolerate higher salt concentrations, but they probably aren't going to grow as well. And so Staph aureus lives on our skin, and our skin can become a little bit salty. And so these organisms are actually able to survive in those conditions. Now, kind of a cool thing that we do, we can use this as a way to select for organisms that might, be able to grow in those environments and differentiate between organisms. So Staph aureus can grow at 7.5% salt. One of the number one ways that we identify Staph aureus is with a agar called mannitol salt agar, which has 7.5% salt concentration. And so they're able to grow there while other organisms like the streptococci cannot. We have our halophiles and our extreme halophiles. You can see that halophiles have this pretty wide range of salt tolerance. So we just talked about Alivibrio fischeri as our microbe of the day. We can see that they're this halophile where they can grow as low as 0. They do pretty well around 5, 6%, and they can even survive up over 10%. These guys are, this is like what you're going to find living in the ocean, which has a salt concentration slightly on this side of the 5%. I think it's like 4. I'd have to look it up. But then we have our extreme halophiles, like this guy Halobacterium solanarum, which can tolerate extremely salty conditions like those found in the Dead Sea and the Great Lakes, the Great Salt Lake. And something to keep in mind is how do you deal with all this salt? So if we remember back to like week 1, if a cell, and these are blood cells, but it works more or less the same for bacteria. If a cell is placed into a hypertonic environment, right? So an environment where there's more osmolytes or more dissolved things outside of the cell than inside of the cell, then as we see in this picture, the water leaves the cell, leading to, in this case, crenation. In the case of bacteria, we'd call it plasmolysis. And then If they're in a hypotonic environment, that means that there's less salt in the environment than there is in the cell. Then we'd see water rush in, which can lead to cell lysis. And so you have to try to maintain an isotonic environment. So if an organism is living in an environment with high salt concentration, how do they maintain an isotonic environment? Because that's the only way that keeps the water from rushing out of the cell like this. So what they end up doing is actually concentrating their osmolytes inside the cell membrane. So we see here, it tells us that the cytoplasm is about 80% water. A normal cell, like we see over here, is actually going to undergo plasmolysis, where the cell membrane and the cell wall pull away from each other, and you get this space. And animal cells, we'd call that prenation, because they don't have a cell wall. And these halophiles are actually going to concentrate all these osmolytes inside of the cell. And by concentrating that organic material and those other salt ions, they're able to maintain that isotonic relationship with their high salt environment. The same thing's going to end up happening in, like, say, for example, a really dry soil. So as soil dries down, you get this relationship where the osmolytes become more concentrated in the water that's left in the soil. And so what the soil bacteria end up doing is actually concentrating osmolytes of their own inside of their cells. That way they don't lose water and go through plasmolysis. All right. So at this point, you should be able to define these terms, optimum, minimum, and maximum, as they relate to our different growth conditions. You should be able to define these different types of temperature preferences, as well as to explain where pathogens fall in this list. And you should be able to define the terms for the different pH environments, acidophilic, alkalophilic, and neutrophilic, and define the what a halophile is and how those microbes combat osmotic stress. So with that, I hope you enjoyed, and I will talk to you soon.